So, good morning, class. This will be the first topic for our semifinals, microbial growth. So, first, let us define your growth. Growth, it is the increase in the amount of cellular components and structure or cell number, which is your population. Your structure is the mass, and your cell number is the number of population. So, the growth of an individual cell is hard to monitor because of the size. So, we have to focus on the growth of the populations, and individual cells also don't affect the environment as much. So, we will focus on the growth of the populations of your microbes. So, prokaryotes generally have two types of growth. First is your binary fission. Second is your budding. So, let's go with binary fission. It is the cell components partitioned equally among two daughter cells. So, I repeat, cell components partitioned equally among two daughter cells. So, that is your binary fission. Most bacteria, example of your binary fission is your unicellular algae and your protozoa. So examples, unicellular algae and protozoa. Next type is the budding. Budding, it is the newly synthesized components which appears only in daughter cells. So, sa budding composed of only one daughter cells, while for binary fission, it composes two daughter cells that is equally partitioned. So, for budding, again, newly synthesized components appear only in daughter cells. So, daughter grows of the parent cell, and the parent can form many daughters. So, good examples of your budding are your yeast and other fungi. So, sa binary fission, example of your binary fission again is your unicellular algae and protozoa. Sa budding naman, example are your yeast and other fungi. So, next slide. What are the methods of growth of your microbes? So first, on a solid medium, individual cells usually gives rise to mass of cells called colony. So first, nagbi-build sila ng colony. Next, we use a solid medium so we can see and manipulate the formed colonies. So, gumagamit tayo ng medium so that we can manipulate the formed colonies. So, one colony is represented by one dot. It is identical to the very first cell unless mutation has occurred. And each species forms colonies of characteristics, shape, size, color, and consistency. So, what are these characteristics? So, first, let us deal with form. So, form meaning what is the basic shape of the colony. So, if we deal with form of the colony, it deals with the basic shape of the colony. Next one is elevation. So, elevation is the cross-sectional shape of the colony. So, elevation, it deals with the cross-sectional shape of the colony. Third is the margin. It magnifies the shape of the edge of the colony. So, it magnifies the shape of the edge of the colony. And fourth, your surface. How does the surface of the colony appear? So, examples of your surface would be smooth, rough, dull, or wrinkled. So, that is the example. And Fifth is your opacity or opacity. Opacity examples are if it's transparent or clear, translucent, iridescent, or opaque. And last is the consistency of the colonies. So, if it is dry or crumbly. So, the question for your consistency, if it is dry or crumbly. So, these are the characteristic shape of your colonies. First is the form, which deals with the basic shape of the colony. Second is elevation, the cross-sectional shape of the colony. Margin, magnified shape of the edge of the colony. Surface, how does the surface of the colony appear? If it's smooth, or rough, or dull, or wrinkled. Fifth, opacity, if it's transparent or clear, opaque, translucent, iridescent. And last is the consistency of the colony, if it is friable, dry, or crumbly. So that will be the methods of the growth of your microbes. So these characteristics are always the same under the same conditions of growing. So again, the size of colony is limited by the exhaustion of nutrients. So the size of the colony is based on the nutrient intake of your microbes. So if larger number of colonies are spread over the surface of the plate, there is not enough space for individual colonies. So this will lead to smaller or exhaustion of the nutrients of the microbes. So next, bacterial cell cycle. So bacterial cell cycle, this is the period in which newly formed bacteria replicate DNA, elongate, and then divide. So bacterial cell cycle, first the bacteria will replicate DNA, and then they will elongate, and after that they will divide. So under optimum conditions, cell divides every 20 minutes, but replication of DNA takes 40 minutes. So this is because DNA never stops replicating. So continuous yung replication ng DNA. So next term would be the population growth. Population growth, first term is the growth rate. So it is the change in cell number per mass per unit time. So it is the change in cell number over mass per unit time. Next is generation, interval for formation of two cells from one. Interval for formation of two cells from one. And generation doubling time. This is the time for one generation to form. So under appropriate conditions, bacteria grow with constant generation time. So example of this is your E. coli or your Escherichia coli, which will always take up approximately 20 minutes to divide. So this allow us to plan experiments in labs, and when we talk about food safety in terms of food poisoning. Okay. Next, after population growth, we will deal with the phases of bacterial cell life. So kindly, ayan. So in the picture, ayan, sa letter B, your exponential growth or your exponential log phase. Exponential log phase. So this will deal with the total cell number doubles with each generation. So total cell number doubles with each generation. So for exponential growth, the increase in cell number can be plotted in a logarithmic scale, which creates a linear scale. So the characteristics of your exponential growth is we'll deal with this. Growth initially slow and then increases rapidly. So sa log phase or sa exponential phase, the growth of the microbes initially will slow down and then they will increase rapidly. So what is the implication of this? The result would be food spoilage or yung pagkasira ng pagkain. So yung mga microbes natin, mabilis silang mag-divide or mag-replicate under your exponential growth. So first initially, they will slow down and then the next will they will increase rapidly. So the implication would be food spoilage. So we have so your population growth cycle, this is the growth of cell population represented by a growth curve. That is your phases of bacterial cell wall, that is the growth curve, which deals with the sample over time to count cells over ml. So let's go with your phases of bacterial cell life or your growth curves. There are 4 phases of curves. 4 phases of curves. Kindly check with that picture. Phases of bacterial cell life. So first phase is your log phase. So log phase, when bacterial cells inoculated into fresh medium, there is a delay before growth starts and cells adapt to the new environment. Also, it is the period of unbalanced growth of your colony or your microbes. This is due to the bacteria's requirement to adjust to the fresh culture. So your bacteria will take time to adjust to the fresh culture or to the new colony. So in your log phase, not all cell components will replicate at the same rate as compared to your exponential phase or your log as in LOG or your exponential phase. So the duration of your log phase depends on the conditions of the culture or your colony. So if you inoculate from exponentially growing culture into the same medium and same conditions, then there will be no delay. So log phase occurs when inoculate old non-growing culture to the same medium, same conditions, and cells are depleted of enzymes. So they need to make more growth before growth can commence. Next, transfer cells from rich growth medium to poorer one, and cells mix enzyme for thing missing in the growth medium. And third, inoculate cells damaged by heat, radiation, chemicals to fresh medium, which will require the time to repair the damage. So, ang iintindihin nyo lang dito sa log phase, it is the period of unbalanced growth. So, the growth of the bacteria will be due to the requirement or how they will adjust to the fresh culture. And also sa log phase, as compared to your exponential phase, mas mabilis ang replication ng bacteria sa exponential phase as compared to your log phase. Okay? Next curve would be the exponential log or your log phase as in log. Ayan. Log. Sa log phase na first is, that is your lag. Sa exponential, or also known as your log phase, that is log. Okay? So, population, doubling at a maximum rate. So, the shortest, this is the shortest generation time. However, mas mabilis ang replication ng bacteria. So, this is a period of balanced growth because all cell components increase at the same rate. And most unicellular microorganisms grow exponentially. And all cell components and the exponential growth rate, it will depend on the culture conditions and characteristics of the organism. However, there is limit which is reached when nutrients runs out and waste accumulates. So, kapag naubos na nila yung nutrients nila, that will be the time na mag-stop yung increase ng rate ng growth nila. So, after that will be your stationary phase, which is your third phase. So, this is the period when nutrients has run out. Ayan, naubos na yung nutrients nila and waste are accumulated. So, there is no net increase or decrease in cell number. So, nag-stop na yung growth ng bacteria. Wala nang increase, wala nang decrease ng growth rate. So, no increase in cell division. Birth rate is equal to your death rate. Ayan. And the last phase would be the death phase. So, at stationary phase, the cells start dying and can lyse. Lyse meaning the cells starts to break down. In your death phase, viable cell count decreases. So, more and more cells dying than being produced. So, your death rate increases as compared to the birth rate. Okay. Next would be dioxic growth. So, your dioxic growth here, there are three definitions or descriptions. Grow cells in presence of two carbon sources. So, what is your two carbon sources? That will be your glucose and your lactose. So, the cells grow in the presence of two carbon sources such as glucose or lactose, and then get two phases of exponential growth separated by transient log phase. So, your enzyme beta-galactosidase is needed to metabolize lactose. So, they need that enzyme beta-galactosidase in order to metabolize the lactose. Okay. The presence of glucose here prevents your beta-galactosidase synthesis, and then the substance that will be broken down will be called as catabolite. So, when glucose is used up, the catabolite repression ends and beta-galactosidase is synthesized. So, after the synthesis of your beta-galactosidase, there will be the presence of your lactose, which will be used as a source of your microbes to grow cells. However, cells that can use uses lactose for growth are better no. Cell grows better on glucose pala. Cell grows better on glucose rather than in lactose. So these are the two carbon sources for your deoxic growth of the bacteria. That is your glucose and your lactose. So next would be the measurement of growth of your bacteria or your microbes. So in measurement of your growth, it is important to determine the population size of your microbes at a specific time. Because it is used to quantify the severity of an infection. Yes. So the measurement of growth of the population size over specific time is used to quantify the severity of an infection. So there are several approaches, all which measure cell number or estimate cell mass. So first measurement is the direct microscopic counts using your hemocytometer. So the hemocytometer counting chamber used with cells in suspension. However, there are limitations in your hemocytometer. Why? Because they are tedious and they does not distinguish death from living cells. Also, it has low sensitivity. So it is tedious. It does not distinguish death from living cells, and it has low sensitivity. The definition of your direct hemocytometer. The hemocytometer counting chamber used with cells in suspension. However, it is limited only. It has limitations because it is tedious to use. It does not distinguish death from living cells, and it has low sensitivity. So next, measurement. Next approach, electron cell counting, or it is also known as your Coulter counters. Coulter counters or electron cell counting. So the limitation here, it is a rapid method, so that is an advantage. However, it can count other particles or dead cells. So it is a rapid method but can count other particles or dead cells. And third, viable cell counts, or that is your plate counting, viable cell counts or your plate counting. It often need to count live cells only. So, the assumption in your viable cells is that one viable cell will produce one colony. One viable cell will produce one colony. And number 4, your optical methods. Optical methods, first, they will determine the turbidity of the colony or the cell culture. The turbidity, if the appearance of the culture is cloudy, or the cell suspension is cloudy or turbid, will cause a light scattered from detecting unit by one cell to be scattered back by another. So, flow cytometry is used to measure DNA. Your optical methods are used to measure DNA, and the presence of your cloudy appearance of cell suspension will mean one unit of cell to be scattered back by another. So the transmission or scattering of a cell is proportional to cell number as well as cell mass. And lastly is the measurement of microbial mass. That is your last approach. Your measurement of microbial mass, it can measure wet or dry weight, and it is used to estimate cell numbers. So it can be used to measure wet or dry weight, and also it is used to estimate cell numbers. So it can also measure mass by determining the amount of biochemical components, the metabolic activity, and the light absorption. So your measurements of your microbial mass can be also measure mass by determining the amount of biochemical components, metabolic activity, and light absorption. So what are the requirements for growth of your microbes? So the requirements for your microbial growth. So first is the supply of suitable nutrients or vitamins. So the supply of suitable vitamins or nutrients. So the raw materials for growth, maintenance, and division are the following: sugars, carbon, hydrogen, oxygen, amino acids, sterols, alcohols, hydrocarbons, methane, inorganic salts, and carbon dioxide. So these are the raw materials for growth, maintenance, and division of your microbial growth or your microbes. So I repeat, sugar, carbon, hydrogen, oxygen, amino acid, sterol, alcohol, hydrocarbons, methane, inorganic salts, and your carbon dioxide. So no individual bacterium can use all possible nutrients. So lack of enzyme, this is due to the lack of enzymes or transport system of your microbes. So they will only have to use one or two or more, but not all nutrients. That will depend on the presence of the enzyme, and transport system for the microbes. So they must have source of carbon and other raw materials for growth, maintenance, and division. So in nature, most bacteria experience periods of starvation, which is this is due to the lack of enzymes or transport system. So most bacteria experience periods of starvation due to lack of enzyme or transport system. So next requirement, wait lang. There are, under your suitable nutrients, there are four nutritional types of or the types of nutritions. So first is your, kindly take this down, photoautotrophs. There are four: photoautotrophs, chemoautotrophs, photoheterotrophs, and chemoheterotrophs. So first, let us deal with photoautotrophs. So the energy source here would be sunlight, and the carbon source is carbon dioxide. So the energy source for photoautotrophs is sunlight, and the carbon source would be carbon dioxide. So the next nutritional type is your chemoautotrophs. The energy source is the inorganic compounds. Inorganic compounds would be the energy source, and the carbon source again will be the carbon dioxide. Third nutritional type is the photoheterotrophs. The energy source would be sunlight. The presence of photo word here would mean sunlight. That is the energy source, and the carbon source would be organic compounds. So the carbon source would be organic compounds. So for chemoheterotrophs, the energy source would be organic compounds, and the carbon source would be organic compounds also. So the energy source and the carbon source for chemoheterotrophs would be organic compounds. Okay. So, ulitin natin. For photoautotrophs, the energy source would be sunlight, and the carbon source would be carbon dioxide. For chemoautotrophs, the energy source would be inorganic compounds, and carbon source would be carbon dioxide. For photoheterotrophs, the energy source would be sunlight, and carbon source would be organic compounds. And for chemoheterotrophs, the energy source is organic compounds, and carbon source would be again organic compounds. So that will be the requirements for your nutrients of growth of the bacteria using suitable vitamins or nutrients. Okay. Next requirement would be the energy source. Energy source. So bacteria have developed general strategies to help cope with any particular shortages. So these shortages, particular shortages, examples would be the decreased production rates for components, low concentration nutrients, or they have utilized alternate terminal electron acceptors during respiration, and obtain nutrients from alternate source. So, the bacteria has employed a specific strategies in order to continue their growth. So, these are they have three specific strategies. So first, highly efficient substrate uptake mechanisms. Second, high surface area to volume. And spore formation. So under your energy source, your bacteria have employed a specific strategies that would mean that in order to continue their growth and to have alternate source of nutrients. So there are three strategies: highly efficient substrate uptake mechanisms, high surface area to volume ratio, and third would be the spore formation. Next requirement would be the appropriate temperature. So the temperature of microorganisms reflects that of their environment. So for every organism, there is a minimum temperature below which no growth occurs. So cytoplasmic membrane must be in a fluid state for proper function. So cytoplasmic membrane must be in a fluid state for proper function. Membrane may gel or freeze, which slows the transport process, so growth cannot occur. So increase in temperature affects organisms in two ways. So the increase in temperature will affect organism in two ways. Chemical and enzymatic reaction occur more rapid, leading to faster growth. So first effect to the organism, the presence of chemical and enzymatic reaction, which would lead to more rapid or faster growth of the bacteria. And at optimum temperature, growth is at its most rapid. So once above maximum temperature, cellular components denature, meaning that growth can't occur. The minimum, optional, optimal, and maximum temperatures are called cardinal temperatures. So first result, chemical and enzymatic reactions leading to faster growth. Next would be in maximum temperature, cellular components will cause denaturation, meaning the growth can't occur, or the growth rate will stop to its maximum rate. Ayan. Next requirement would be the presence of water. Ang gulo. Water. So all organisms require water for life. So it makes up roughly 80% of the bacterial mass. So your water will contain 80% of the mass of the bacteria. During growth, nutrients enter and waste products and leave all in solution, meaning in the presence of water. Bacteria can grow only in or on materials that have adequate or available or free water. So the availability of water to microorganisms depend on the water content of the environment, the water adsorption to surface, the solute dissolution in water. So again, availability of water to microorganisms depends on the water content of the environment, water adsorption to surface, and the solute dissolution in water. So this relates to vapor pressure of water in air over a surface. So remember that microbes can transport water. So it flows in or out of the cell via osmosis. So the process used by the bacteria in order to transport water is osmosis. And last requirement, the appropriate levels or absence of oxygen and pH. So first, let us deal with pH. pH is the power of hydrogen ion concentration. So again, pH is the power of hydrogen ion concentration. Microbes often change pH as they grow. So they have buffers to keep pH in certain range. For example, your acidophiles will grow at low pH. Examples of your acidophile would be fungi. Some bacteria are obligate acidophiles. They are unable to grow at pH of 7. So, acidophiles from the word acid, meaning their growth would be at a low pH or lower than 7. So they are unable to grow at pH 7. So the membrane dissolves, indicating high hydrogen ion concentration is needed for membrane stability. So that is the first type of your microbes under your pH. Next, alkaliphiles. Alkaliphiles from the word alkali. So, bacteria found in highly basic habitats. So, they will depend at pH higher than 7. So, the internal pH is probably near neutral since many cellular substances are acid or alkali liable. So, organisms can modify pH in several ways. So, there are 4 ways for organisms to modify their pH. First, acid production by fermentation. Acid production by fermentation, such as glucose to lactic acid. Acid production by fermentation, such as the process of glucose into producing a lactic acid. Next, excretion of hydrogen ions into medium. Excretion of hydrogen ions into medium. Production of ammonia. Production of ammonia due to the deamination of amino acids. Deamination of amino acids. Production of ammonia. And lastly, selective removal of nutrients. So the increased pH by taking up ammonia, lower by taking up nitric acid. So they will increase their pH by taking up ammonia, or they will lower their pH by taking up nitric acid. So the presence of ammonia in microbial growth will increase the pH, while the presence of nitric acid in microbial growth will decrease the pH of the microbes. Next is the presence of, last requirement would be the presence of oxygen. So 20% of gas in atmosphere is oxygen. I repeat, 20% of gas in atmosphere is oxygen. And the use of oxygen as an energy source involves your redox reaction. Redox meaning reduction, oxidation, reaction. So more reduced a compound is, the more extra electron it carries, the more energy it contains, the greater the tendency to donate electrons. So organism can use redox reaction in order to extract energy from nutrient molecules. So the presence of your redox reaction would give way to extraction of nutrient molecules by the microbes. So microbes that use oxygen are designated aerobes. So microbes that use oxygen are designated aerobes. There are 3 types. Your obligate aerobes. Obligate aerobes, please take this down. This will require oxygen in the biosynthesis of sterols and unsaturated fatty acids. So your obligate aerobes will require oxygen in the biosynthesis of sterols and unsaturated fatty acids. Next is your facultative aerobes. These do not require oxygen but grow better with it. So these do not require oxygens but grow better with it. So your facultative aerobes, the presence of or absence of your oxygen does not affect their growth rate. However, in the presence of oxygen, they grow better with it. Okay? For microaerophilic, that is your last aerobes, it requires oxygen but at levels lower than atmospheric. So it requires oxygen but at levels lower than atmospheric. Some microorganisms can't use oxygen as a terminal electron acceptors and they are called your anaerobes. So yung na-discuss natin kanina are the aerobes. In here naman, they are anaerobes. So the definition of your anaerobes, some microorganisms can use oxygen as a terminal electron acceptors. There are 3 again, aerotolerant anaerobes, aerotolerant anaerobes. They grow in the presence of oxygen but do not use it. So aerotolerant anaerobes, they grow in the presence of oxygen but don't use it. For obligate anaerobes, obligate anaerobes. So for obligate anaerobes, they are killed by oxygen. So the presence of oxygen will cause them to deteriorate. Last, sensitivity of obligate anaerobes to oxygen will reflect the inability to neutralize toxic products of oxygen metabolism. So that will cover the topics of microbial growth.